Hi students, this is A. Mandari Kumar Rajesh. In this video, we are going to discuss the remaining topics which are in the structural chromosomal aberration or the structural chromosomal mutation. In the last video, we have discussed up to the changes in the number of gene loci, and it has two types. We discussed the two types. Then in this video, we are going to discuss the remaining topic, changes in the arrangement of gene loci, and it has two types. They are classified into two types. They are inversion and translocation. Then this inversion process again classified into two types. They are paracentric inversion. They are paracentric inversion, then pericentric inversion. Pericentric inversion. Then the translocation again divided into three types. The translocation process divided into three types. They are simple translocation, shift translocation, then the third one is reciprocal translocation. Then the reciprocal translocation again classified into two types. They are the reciprocal translocation again divided into two types. They are homozygous translocation and heterozygous translocation. Now I am going to explain inversion first and their types. Next I am going to explain the translocation process and their types. So please wait a minute. Let me teach you the process inversion. Inversion means the rearrangement of order of genes in a chromosome by reverse by an angle 180 degree. So the inversion means rearrangement of order of genes in a chromosome by reverse by an angle 180 degree. Then this process involves two chromosomal breaks and reunions. Takes place. Then during this process there is neither gain nor loss. That means there is no loss and there is no gain, but the gene sequence is rearranged during inversion. Process. So gene sequences rearranged during inversion process. Then the inversion process was first reported in the fly Drosophila. And it was reported by the scientist Sturtevant in the year 1926. And the inversion may divided into two types. They are paracentric inversion. Then the second type is pericentric inversion. Paracentric inversion means an inversion takes place apart from the centromere. While in the case of pericentric, an inversion includes the centromere. Pericentric means an inversion includes the centromere. So now I am going to explain the paracentric with a diagram. So it is a chromosome, normal chromosome, which contains the gene sequence capital A, capital B, capital C, capital D, capital E. Here F, G, H, I. So in this chromatid, in this arm, mutation takes place in the capital B, capital C and capital D. So in the three genes, the mutation, that inversion occurs. The inversion occurs. Then the gene sequence are rearranged like this. So that chromosome contains, the mutated chromosome contains capital D, capital C, then capital B, here capital E, then capital F, capital G and capital H, capital I. Here in the chromosome two breaks occurs. So here, so in the inversion process two breaks and reunions occurs. So here two breaks occur and two reunion process occurs. Then the capital B comes near to the centromere, then the capital D goes near to the terminal portion. So inversion process takes place in the chromosome, and that mainly paracentric inversion takes place in the chromosome. That means the centromere is not involved in this inversion. The inversion takes place apart from the centromere, and that process is called paracentric. Then pericentric inversion. So this is a chromosome which contains capital A, capital B, capital C, capital D, then capital E, then capital F, capital G, capital H, capital I. So in this chromosome, the inversion occurs on capital D, capital E and capital F genes. So the pericentric inversion, that mutation occurs on capital D, capital E, capital F. In pericentric inversion, the arrangement of gene sequences rearranged. And in pericentric inversion, centromere also involves. Centromere also included in the pericentric inversion. So the pericentric inversion takes place and rearrangement of sequence also takes place. So capital A, capital B, capital C, then capital F, then the long segment, the long arm contains the capital E and capital D, then capital G, capital H and capital I. So due to pericentric inversion, the centromere also involves and rearrangement of gene sequence takes place. So that's all about the inversion process. Now I am going to explain the remaining process, translocation and its types. Let me explain the remaining process, translocation. Translocation means the exchanges of the segments of chromosome to a non-homologous chromosome is called as translocation. Translocation means the exchanges of segment of chromosome to a non-homologous chromosome. Then don't have confusion with the crossing over process and the translocation process. Crossing over means the exchanges of genetic material between two homologous chromosomes, while in the case of translocation, the chromosomes are interchanged to a non-homologous chromosome. In the translocation process, the exchanges of chromosome segments between two non-homologous non-homologous chromosome. Then crossing over leads to genetic variation, while in the case of translocation, it leads to genetic abnormality and it leads to mutation. So that is the difference between the crossing over and translocation. Then translocation can be divided into three types. They are simple translocation, then shift translocation, then reciprocal translocation. Simple translocation can be defined as, in this translocation, a single break is occur on one chromosome. And that broken segment is attached to one end of non-homologous chromosome, and that broken segment of that chromosome attached to one end of non-homologous chromosome, and it is a rarely occurring process. The simple translocation process is a rare occurring process. So now I explain with a diagram. So it is a chromosome and it is a non-homologous chromosome to this chromosome. This chromosome is non-homologous to this chromosome. Then it contains A, here it contains B, it contains C, it contains D. So the segment is broken here, the segment is broken in this chromosome, and it is attached to the one end of non-homologous chromosome, and this segment is attached to one end of non-homologous chromosome, and that process is called as simple translocation, Then shift translocation means the broken segment of chromosome get inserted middle of non-homologous chromosome. The broken segment of chromosome is inserted middle of non-homologous chromosome. For example, so this is a chromosome and this is non-homologous chromosome to this chromosome. Then capital A, capital B, capital C, then capital D, capital E, capital E. Here the broken segment, so suppose the translocation process occurs in the capital A. So that segment is breakdown and that segment is broken down, and they are inserted in the middle of non-homologous chromosome, and they are shifted to middle of the non-homologous chromosome, and that process is called as shift translocation, Then the final one is reciprocal translocation. So reciprocal translocation may be divided into homozygous, then heterozygous translocation, homozygous and heterozygous translocation. Then reciprocal translocation may be defined as exchange of chromosome segment between two non-homologous chromosomes. That means the mutual exchange of the segments of chromosome between two non-homologous chromosomes. So it is otherwise called as illegitimate crossing over. The reciprocal translocation is otherwise called as illegitimate crossing over. Now I am going to explain the two types, homozygous translocation and heterozygous translocation. Here I have drawn the diagram of chromosomes. This diagram shows two pairs of normal homologous chromosomes. This diagram shows two pairs of normal homologous chromosomes. Then I am going to explain the homozygous translocation. Homozygous translocation. In homozygous translocation, both the chromosome of two pairs are involved in translocation. Both the chromosome of two pairs of chromosomes are involved in translocation. Then two homologous of each translocated chromosomes are identical. Two homologous of each translocated chromosomes are identical. So don't get confused. I will tell you clearly. So it is a pair of homologous chromosome and it is a pair of homologous chromosome. These two pair of homologous chromosomes are involved in translocation. Involved in the translocation. That is why this process is called homozygous translocation. Homozygous translocation. Then you can find the two homologous chromosomes are identical. You can find the two homologous chromosomes are identical, and here also you can find the two homologous chromosomes are identical. So the translocation occurs and each homologous translocated chromosomes are identical, and that process is called as homozygous translocation, and that process is called as homozygous translocation process. In heterozygous translocation, only one of the chromosome is involved in translocation process, and that chromosomes are come from each homologous pairs of chromosome. So here it shows two pairs of homologous chromosome. So only one of the chromosome is involved in the translocation process and the remaining one chromosome is in normal condition. So here you can find two pairs of homologous chromosome. So in the translocation process, only one chromosome from each pair of homologous takes place. Only one chromosome from each pair of two homologous takes place in the translocation process, and the remaining one chromosome is normal in condition. So here you can find this chromosome in normal condition, and this chromosomes involved in this chromosome is involved in translocation process, and in this pair of homologous chromosome, this chromosome is in normal condition and this chromosome involved in translocation process, and that process is called as heterozygous translocation, and that process is called as heterozygous translocation. Then translocation process leads to the production of new species. Translocation process leads to the production of new species. So that's all about today's video. If you have any doubt, please let me know. Thank you very much for watching this video. Thank you.